start talking about muscular tissue, which I think is a little unusual because last week we learned about the muscular system, and a big part of muscular tissue is skeletal muscle, but this is the way it's set up. So all muscles are excitable. They can respond to things like chemical signals, stretch, electrical changes across the plasma membrane, and very closely related to that, they are conductive. Local electrical changes on the cell lead to greater waves of excitation that travel along the entire muscle fiber. Muscle cells are contractile. They have the ability to get forcibly shorter when they are stimulated, because of their excitability and conductivity. In addition to that, they are also extensible. They can be stretched between contractions, and they are elastic. They can return to their original length after being stretched. Skeletal muscle, the first of the three, is a voluntary striated muscle, and it's usually attached to bones. We know from last week some of the muscles are attached to the fascia under our skin and our face, but most are attached to bones. They have those striations, the alternating light-dark transverse bands, because of the arrangements of their actin and myosin proteins. They are voluntary, which means they are subject to conscious control. The other types of muscle are involuntary. Now, don't get reflex actions confused with voluntary. They average about 100 microns in diameter and about 3 centimeters long. However, they can be much thicker than that, 500 microns, and as long as 30 centimeters. Skeletal muscles also are called muscle fibers or myofibers. Here we have an image of a skeletal muscle fiber. You can very nicely see the striations in this image. You can see that most of them are multinucleated, and they also contain fibrous connective tissues. There's an endomysium surrounding each single muscle fiber or cell. There's perimysium that bundles those fibers into fascicles. And then there's epimysium that surrounds the entire muscle. And a big part of that fibrous connective tissue is collagen, which is somewhat extensible and elastic, meaning it will stretch slightly under tension and recoil when released. That's going to help the muscle resist excessive stretching and protects the muscle from injury. Also helps return the muscle to its resting length. Is going to contribute to the power output and the efficiency of each of the muscles. A single muscle fiber or cell, again, is surrounded by a plasma membrane, as all cells are, and in the case of a muscle cell, we call that plasma membrane the sarcolemma. There is also cytoplasm inside of the muscle cell, and in the case of a muscle, we call it sarcoplasm. We have myofibrils, the long protein cords occupying most of the sarcoplasm. Glycogen is found there. Glycogen is a carbohydrate storage form of glucose. Myoglobin is a relative of hemoglobin. It is a red pigment. It can store oxygen for muscles to use when they are active. They have many mitochondria. They are a powerhouse of the cell producing ATP, and the best way to contract the muscle is using oxidatively phosphorylated obtained ATP. Again, they have multiple nuclei. They average between 30 and 80 per millimeter. DNA instructions are in each one of those nucleus, so the more you have, the more branches you have of that library, the more easier it is to get instructions where they need to go. They also have endoplasmic reticulum, and most of the endoplasmic reticulum in a muscle cell is smooth endoplasmic reticulum, and it's got a different function than it does in most cells. So we call it sarcoplasmic reticulum. And that sarcoplasmic reticulum also contains terminal cisterns, which are dilated sacs that cross the muscle fiber from one side to another. And the major purpose of the sarcoplasmic reticulum is to serve as a calcium reservoir. It's going to release calcium through channels when activated. There's a protein within the sarcoplasmic reticulum called calsequestrin. There are also transverse tubules. They are infoldings of the sacrolemma that penetrate through the cell and emerge on the other side. Each T-tubule is going to have two terminal cisterns associated with it on either side, and these play a role in getting signals throughout the cell. Myoblasts are stem cells that fuse to form each muscle fiber early in development. Each one of them would contribute a single nucleus. There are satellite cells that are unspecialized myoblasts remaining between the muscle fibers and endomysium, and they play a role in regenerating damaged skeletal muscle tissue. Here is our diagram of a skeletal muscle cell. Myofibrils are the muscle filaments or myofilaments. There are three kinds of filaments. The thick filaments are made of several hundred myosin molecules. Myosin, remember, is a motor protein. Each molecule is shaped like a golf club. Two chains intertwine to form a shaft-like tail and a double globular head. Those heads direct outward in a helical array around the bundle. The heads on one half of the thick filament angle to the left, while the others angle to the right. There is a bare zone with no heads in the middle. So there is our intertwined myosin molecule. See it as a cluster, and we get the myosin head. The thin filaments are composed of three different proteins. There is fibrous actin, which are two intertwined strands of globular actin, each with an active site that can bind the head of myosin. There is tropomyosin. It blocks 6 or 7 active sites on the G-actin subunits. And troponin, which is a small calcium-binding protein on each tropomyosin molecule. And these are going to be important in how contraction occurs. Here's our thin filaments. So when we put the two together, the thick filaments and the thin filaments, we have the bare zone in the middle with the myosin heads and the actin filaments up above them, or on the other side of them, really. There are also elastic filaments. They're a huge springy material called titin. It runs through the core of the filament, anchors to the Z disc or an M line, and that's going to help stabilize the position of the thick filament, and it prevents overstretching, and additionally it will provide poor recoil. Of the myofilaments, myosin and actin are the contractile proteins. They do the work of contraction. Tropomyosin and troponin act as regulatory proteins. They act like a switch that determines when the fiber can be contracted. Contraction is activated by the release of calcium into the sarcoplasm from the sarcoplasmic reticulum and binding to troponin. Troponin, with calcium bound to it, changes shape and moves the tropomyosin away from the active sites, the binding sites for myoglobin. There are several other proteins associated with the myofilaments that anchor, align, and regulate them. One of them is dystrophin. Dystrophin links the actin in the outermost filaments to the membrane proteins that link it to the endomysium. This transfers the forces of muscle contraction to the connective tissue, ultimately leading to the tendon. It is genetic defects in dystrophin that produce muscular dystrophy, Duchenne's muscular dystrophy. And this is kind of lining them all up schematically. In cardiac and skeletal muscles, myosin and actin are organized in a precise array that accounts for the striations. The striations are alternating A and I bands, dark A, light I. The A, or dark band, is the darkest part where the thick filaments overlap with a hexagonal array of thin filaments. There is a H band in here as well that is not as dark. It's in the middle of the A band and contains thick filaments only. There's an M line, which is a dark transverse protein in the middle of the H band. The I band, the light band, has the Z disc or Z line, which is a protein complex providing anchorage for the thin filaments and elastic fibers. And I would encourage you to go to the text and look at these images and really get the explanation from the text, because this organization of contractile proteins is really the key to how both cardiac and skeletal muscles will contract. The sarcomere is the segment from Z disc to Z disc. That would be the functional contractile unit of the muscle fiber. Muscle cells shortened because their individual sarcomeres shorten. The Z discs are pulled closer together as thick and thin filaments slide past each other. Neither the thick nor thin filaments change length during the shortening. Only the amount that they overlap is going to change. And during that shortening, dystrophin and other linking proteins pull on the extracellular proteins, transferring that force to the extracellular tissue. Remember, these are only cells. Skeletal muscle can't contract unless it's stimulated by a nerve, and if a nerve connections are severed or poisoned, the muscle is paralyzed and may waste away. It's called denervation atrophy. Skeletal muscles are served by somatic motor neurons. These are nerve cells whose cells body are in the brain stem and spinal cord that serve the skeletal muscles. Their axons, called somatic motor fibers, lead to the skeletal muscle. Each nerve fiber will then branch out to a number of muscle fibers, so one nerve serves several muscle cells, and each muscle fiber is supplied by only one motor neuron. So one motor neuron covers several cells, but each cell is by an individual motor neuron. So that motor unit, it would be the one nerve fiber and all the muscle fibers that are innervated by it. That will behave as a functional unit. Muscle fibers of one motor unit are dispersed throughout the muscle. They will contract in unison. They can produce weak contractions over a wide area. They provide the ability to sustain long-term contractions as the as the motor units will take turns contracting. An effective contraction usually requires the contraction of several motor units all at once. An average motor unit contains 100 muscle fibers distributed across as many as 100 different fascicles. We have various sized motor units. Small motor units provide a fine degree of control. So, for example, in the eye and the hand, there are muscles that are controlled by motor units with only 3 to 5 muscle fibers in each. Large motor units are going to provide more strength than control. Powerful contractions will be supplied by large motor units with hundreds of fibers. So, for example, in our quadriceps femoris and our gastrocnemius are controlled by motor units that may have 1,000 muscle fibers. The place where the nerve and the muscle actually very close to come in contact, since they don't actually touch, is called the neuromuscular junction. It's an example of a synapse, a point where a nerve fiber meets its target cell. When the target cell is a muscle fiber, we also call it a neuromuscular junction or a motor end plate. Each terminal branch of the nerve fiber within the neuromuscular junction forms a separate synapse with the muscle fibers. That will consist of the axon terminal, the slightly swollen end of the nerve fiber. That nerve fiber on that end has synaptic vesicles filled with neurotransmitter, and in the case of skeletal muscles, that neurotransmitter is acetylcholine. The nerve impulse will cause the synaptic vesicles to undergo exocytosis that will release acetylcholine into the small gap between the muscle cell and the nerve cell called the synaptic cleft. And that synaptic cleft will then be filled with acetylcholine. The sacrolemma has millions of acetylcholine receptors, which are proteins that respond to acetylcholine. The postsynaptic membrane folds or junctional folds of the sacrolemma increase surface area, maximize the number of acetylcholine receptors there, and the lack of receptors can cause a weakness in the disease myasthenia gravis. The entire neuromuscular junction is enclosed in basal lamina, which is a thin layer of collagen and glycoproteins separating the muscle fiber and the nerve ending from the surrounding connective tissue. It passes through the synaptic cleft and nearly fills it. The sacrolemma and the lamina with the synaptic cleft contain acetylcholinesterase, an enzyme that will break down acetylcholine, and that will allow for relaxation of the muscle. Muscle fibers and nerve cells are both electrically excitable cells. Their membranes exhibit voltage changes in response to stimulation. This would be electrophysiology if you studied the electrical activity of cells. The electrical activity is going to depend on the concentration differences in ions in the intracellular fluid and the extracellular fluid. Intracellular fluid has a greater concentration of negative ions. Negatively charged proteins account for most of them, in addition to nucleic acids and phosphates. And the extracellular fluid is more potassium ions, more positive ions. So in a resting state, that membrane is polarized. It has the inside has a net negative charge. Any difference between charges in 2 points is called an electrical potential, or measured as volts. So in an unstimulated resting cell, the plasma membrane is electrically polarized. It's charged with a negative resting membrane potential. In a stimulated cell, the ion channels in the membrane are going to open, allow sodium to flow into the cell, which would be down its electrochemical gradient, and that causes depolarization of the membrane. Then additional channels allow potassium to flow out of the cell, which will repolarize the membrane. And this up and down voltage shift is what we call an action potential. There are some toxins with synaptic functions that can paralyze muscle cells. Some pesticides contain cholinesterase inhibitors. They will bind to acetylcholinesterase and prevent it from degrading the acetylcholine. That can lead to spastic paralysis, continual contraction of the muscles. Tetanus or lockjaw is a form of spastic paralysis caused by the toxin Clostridium tetani. Here, glycine in the spinal cord normally stops motor neurons from producing unwanted muscle contractions, but the tetanus toxin blocks glycine release in the spinal cord and causes overstimulation and spastic paralysis of the muscles. Flaccid paralysis is a state in which the muscles are limp and cannot contract. There's a poison called curare that competes with acetylcholine receptor sites but doesn't stimulate the muscle. It's derived from plants used by South American natives to poison blowgun darts. Botulism is a type of food poisoning caused by a neuromuscular toxin secreted by the bacterium Clostridium botulinum. It blocks the release of acetylcholine, causing flaccid paralysis. Botox cosmetic injections are used for wrinkle removal. There are 4 major phases of contraction and relaxation: excitation, contraction, coupling, contraction, and relaxation. In excitation, action potentials in the motor nerve lead to action potentials in the muscle fiber. In excitation, contraction, coupling, events that link the action potentials on the sacrolemma to activation of the myofilaments, preparing them to contract. Contraction are the steps in which the muscle fiber develops tension and may shorten. And relaxation, when stimulation ends, a muscle fiber relaxes and returns to its resting length. And I'm going to show you a brief video explaining that better than I can. I hope.
So what is ClickUp? ClickUp is an all-in-one productivity software where you can manage everything and anything related.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay, let's see.
Hi, I'd like to...
It's probably a good video if you can find it on YouTube, but not part of this lecture. So there's a length-tension relationship. The amount of tension generated by a muscle depends on how stretched or shortened it was before it was stimulated. If it's overly shortened before stimulation, we get a weak contraction because the thick filaments already butt up against the Z-disc. If it's too stretched before stimulation, we also get a weak contraction because we get minimal overlap between thick and thin filaments, which results in minimal cross-bridge formation. So optimum resting length produces the greatest force when the muscle contracts. That's why, one of the reasons why the nervous system maintains muscle tone, a partial contraction, ensuring that resting muscles are near this length. Rigor mortis is a hardening of the muscles, stiffening in the body that begins about 3 to 4 hours after death, typically. After that, the deteriorating sarcoplasmic reticulum will release calcium, and the deteriorating sarcolemma allows the calcium to enter the cytosol. That calcium activates myosin cross-bridge formation and the muscles contract. Muscle relaxation requires ATP, and we don't have ATP being produced after death, so the fibers remain in a contracted unit until the myofilaments begin to decay. Rigor mortis tends to peak approximately 12 hours after death and then diminishes over the next 48 to 60 hours. Muscle twitch is a quick cycle of contraction and relaxation when a muscle is directly stimulated by an electrode. A muscle twitch is not like a twitch we have physiologically. It's a way to study the contraction process. A myogram is a graph that measures muscle twitch. It shows the three phases: the latent period, the contracting phase, extension, tension, and the relaxation phase. That latent period is a delay just after stimulation of the muscle, the time required for the excitation, contraction, coupling, and tensing elastic components. It generates internal tension, but it's not visible movement on the myogram. The contraction phase and external tension is generated and the load is moved as the muscle fibers shorten. And in the relaxation phase, the sarcoplasmic calcium levels fall because calcium will be reabsorbed into the sarcoplasmic reticulum and the tension will decline. And that is shown in this graph. There is a threshold. There is a minimum voltage that causes a muscle to twitch. So even if the same voltage is delivered, different stimuli cause twitches varying in strength and length. Varying in strength because the muscle's starting length influences tension generation. Muscle fatigue after continuous use. Warmer muscle enzymes work more quickly. Muscle cell hydration levels influence cross-bridge formation. Increasing the frequency of stimulation delivery increases tension output. Muscles must contract with variable strength and for different tasks. You hold a glass differently than you hold a hammer. Stimulating the nerve with higher voltage produces stronger contractions. Higher voltages excite more nerve fibers, which stimulate more motor units to contract. So we can have recruitment or multiple motor unit summation, which is a process of bringing more motor units into play with stronger stimuli. It occurs according to the size principle. Weak stimuli, which means low voltage, recruit smaller units, while strong stimuli recruit small and large units for more powerful movements. Frequency of stimulation also affects twitch strength. Low frequency stimuli produce identical twitches. Higher frequency stimuli, 20 stimuli per second, produce temporal or wave summation, because each new twitch is going to ride piggyback on the previous one, generating higher tension. What it really means is we get partial relaxation between stimuli and the next wave. Call that incomplete tetanus. Unnaturally high stimulus frequencies in lab experiments cause a steady contraction called complete or fused tetanus. And that's shown here on the third graph. My arrow's gone, this cursor's gone. There are different kinds of contractions. We can have an isometric contraction, is a contraction without a change in length. Muscles produce internal tension, but external resistance caused it to stay the same length. That can be a prelude to movement when the tension is absorbed by elastic components of the muscle. It is also important in postural muscle function and antagonistic muscle joint stabilization. We have isotonic contractions, which are contractions which change the length, but no change in the tension. So the muscle changes in length with no change in tension because the muscle gets shorter as it maintains tension. So if we are lifting a weight, we can also have eccentric contractions. If the muscle lengthens as it maintains tension, and so when you are setting down those weights, those are both forms of contraction. In an isotonic contraction, we get contraction with a change in length but no change in tension. They can be concentric or eccentric. So in a concentric, the muscle will shorten, maintaining its, we just went over this, did we not? Yes, we did. My apologies. So here, isometric contraction, isotonic contraction, and isotonic concentric contraction, and isotonic eccentric contraction. All muscle contractions depend on ATP. That ATP supply depends on the availability of oxygen, glucose, fatty acids. There are two main pathways of ATP synthesis. The anaerobic fermentation enables cells to produce ATP in the absence of oxygen. We get very little ATP comparatively, and we get a substance called lactate, which needs to be disposed of by the liver. In aerobic respiration, we get far more ATP. We don't generate lactate, and we get but we must have a continual supply of oxygen. So our immediate sources for short, intense exercise, oxygen briefly supplied by the myoglobin but rapidly depleted. Muscles meet most of their ATP demand by borrowing phosphate groups from other molecules and transferring them to ADP. Two enzymes control the phosphate transfers, myokinase and creatine kinase. They will both transfer a phosphate to an ADP. The phosphagen system is the combination of ATP and creatine and phosphate, which provides nearly all the energy for short bursts of activity, which is approximately 6 seconds of sprinting, for example. As the phosphagen system is exhausted, muscles must shift to anaerobic fermentation. They will obtain glucose from the blood and their own glucose. They will get a net gain of 2 ATP, but each glucose molecule will produce 2 molecules that are converted into a 3-carbon lactic acid. The anaerobic threshold, or the lactate threshold, is the point at which lactate becomes detectable in the blood. Glycogen lactate synthesis is the pathway from glycogen to lactate. Produces enough ATP for 30 to 40 seconds of maximal activity. Long-term energy, after about 40 seconds, the respiratory and cardiovascular systems will start to deliver oxygen fast enough for aerobic respiration to meet most of the muscle's ATP demands. So here we have aerobic respiration produces ATP, then glycolysis, approximately 36 ATPs instead of 2. The efficient means of meeting the ATP, it's the only efficient means of meeting the ATP demands for prolonged exercise. After about 3 or 4 minutes, the rate of oxygen consumption levels off, so we get to a steady state where aerobic ATP production keeps pace with demand. And for 30 minutes or so, energy comes equally from glucose and fatty acids. Beyond 30 minutes, the depletion of glucose causes the fatty acids to become the more significant fuel. Muscle fatigue is a progressive weakness from prolonged use of the muscles. Fatigue and high-intensity exercise is thought to result from potassium accumulation in the T tubules, ADP and inorganic phosphate accumulation. Fatigue and low-intensity or long-duration exercise is thought to result from fuel depletion as we run out of glucose and glycogen, electrolyte loss through sweat, and central fatigue, where less motor signals are issued from the brain, inhibited by the exercising muscles release of ammonia. Our maximum oxygen uptake is the point at which the rate of oxygen consumption plateaus and does not increase further with added workload. It is the major determinant of one's ability to maintain high intensity exercise for more than 4 to 5 minutes. It will be proportional to your body size and peaks around age 20. It's usually greater in males than females. It can be twice as great in trained endurance athletes as in an untrained person. Excess post-oxygen consumption, used to be referred to as oxygen debt, is the elevated rate of oxygen consumption following exercise that aerobically replenishes ATP. It replaces oxygen reserves on myoglobin, provides oxygen to the liver, getting rid of lactate, provides oxygen to many cells that have elevated metabolic rates after exercise. EPOC can be 6 times the basal consumption, and it can last for up to an hour after exercising. Skeletal muscle fibers are classified into two major physiological classes: slow twitch, slow oxidative, red fibers, type 1 fibers. These are well adapted for endurance, resist fatigue by oxidative ATP production. They're important for muscles that maintain posture. They're thin cells with abundant mitochondria, capillaries, and myoglobin, and contain a form of myosin with a slow ATPase and sarcoplasmic reticulum that releases calcium slowly. They can be grouped into small motor units controlled by small, easily excited motor neurons that allow for precise movements. The fast twitch, fast glycolytic fibers, or white fibers, type 2 fibers, are well adapted for quick response utilizing glycolysis and anaerobic fermentation for energy. They're abundant in quick, powerful muscles, the eye, hand muscles, the gastrocnemius. They contain a form of myosin with fast ATPase and a large sarcoplasmic reticulum that releases calcium quickly. Their fibers are thick and strong. They have low myoglobin levels, giving them a pale color. They're grouped into large motor units controlled by larger, less excitable neurons, allowing for powerful movements. We also have intermediate or fast oxidative fibers. It's mainly in other mammals, but relatively rare in humans. And here you can see that an individual fascicle will have some of each. Every muscle contains a mix of fiber types, but one type tends to predominate depending on the muscle function. Fiber type within a muscle will differ between individuals. Some individuals seem genetically predisposed to be sprinters, while others are more suited for endurance. Factors that affect muscle strength include muscle size. A thicker muscle forms more cross bridges, and the muscle can exert a tension of 3 or 4 kilograms per centimeter square. Fascicle arrangements: pennate are stronger than parallel. Parallel are stronger than circular. Size of active motor units: the larger the motor unit, the stronger the contraction. Multiple motor unit summation: simultaneous activation of more motor units should increase tension. Temporal summation: the greater frequency of stimulation, the stronger the muscle contraction. The length-tension relationship: a muscle at resting optimal length is prepared to contract more forcefully than a muscle that is excessively contracted or stretched. Fatigue: fatigued muscles contract more weakly than rested muscles. There are different types of exercises that have different effects on muscles. Resistance exercise: contraction of a muscle against a load that resists movements, weightlifting, for example. Here we get growth in cellular enlargement. Muscle fibers synthesize more myofilaments and myofibrils and grow thicker. We also have endurance or aerobic exercise, which we get contraction of the muscle against a load that resists movements, for example, lighter weightlifting. This will improve the fatigue and resistance of muscle, tends to increase the skeletal length, red blood cell count, and oxygen carrying capacity of the blood. It also enhances cardiovascular, respiratory, and nervous system function. Cardiac and smooth muscles share certain properties. Their cells are myocytes, but they are not long and fibrous as they are in skeletal muscles. In fact, they tend to be relatively short and only have 1 or 2 nuclei. Cardiac muscle cells are specifically called cardiomyocytes. They are involuntary. They are not under conscious control. A conscious mind can affect the rate, but they can't start it or stop it. They receive innervation from the autonomic nervous system as opposed to somatic motor neurons. Cardiac muscle cells contract with a regular rhythm. They work in sleep and wakefulness without fail and without conscious attention. They have a high resistance to fatigue. The muscle cells of a given chamber must contract in unison, and the contractions must last long enough to push the blood out of the heart. The cardiac muscle cells are striated, relatively short and thick, surrounded by endomysium, but not peri or epimysium. Cardiomyocytes branch and join into other cells called intercalating discs. There are no satellite cells and very limited mitosis. Cardiac muscle cells tend to be replaced by fibrous connective tissue. They can contract without the need for nervous stimulation. They have built-in pacemaker that rhythmically sets off a wave of electrical excitation. Waves travel through the muscle and triggers contraction of the heart chambers. The heart is autorhythmic. It's able to contract rhythmically and independently. And it's highly resistant to fatigue. Smooth muscle, the structure and function of smooth muscles, they are named for their lack of striations. Their innervation varies. Some smooth muscles lack nerve supply, others receive input from automatic fibers with many varicosities containing synaptic vesicles. They are capable of mitosis and hyperplasia. Injured smooth muscle cells regenerate relatively well. Contraction is slower than skeletal muscle and cardiac muscle. It takes longer to contract, but can remain contracted for a longer time without fatigue. They form the layers within walls of hollow organs so they can propel contents of the organ and can modify pressure and flow of blood in the circulatory system. They can provide fine control in some locations. For example, the smooth muscle of the iris controls the pupil size. Piloerector muscles raise the hairs in the skin. They have a fusiform shape, wide in the middle, tapered at the ends. Single nucleus located near the middle of the cell. They have thick and thin filaments present, but they are not aligned with each other, so we don't get the striations. It's still actin and myosin, however. They have minimal sarcoplasmic reticulum. They do not contain T-tubules. Their calcium comes from extracellular fluid by way of channels in the sarcolemma. The Z discs are absent. They are changed with dense bodies. And those dense bodies are protein masses that form plaques on the inner face of the plasma membrane and are scattered throughout the sarcoplasmic. There are multi-unit smooth muscle cells and unitary or single-unit smooth muscles. Individual myocytes respond independently of the others in a multi-unit. In a unitary, the myocytes are coupled by gap junctions, so they contract as a unit. Smooth muscle can contract or relax in response to a variety of stimuli. The autonomic nervous system has nerve fibers and neurotransmitters. Parasympathetic nerves secrete acetylcholine, stimulating the GI tract, smooth muscle, and sympathetic nerves secrete norepinephrine, which relaxes smooth muscle in the bronchioles. There are chemicals that they will respond to: low oxygen, hormones, pH, temperature. Cold excites piloerector muscles and warmth relaxes the muscle. Stretch, the stomach contracts when stretched by food, and many are autorhythmic, such as many of those in the digestive tract. Contraction is always triggered by calcium, energized by ATP, and achieved by sliding filaments. Again, smooth muscles get most of their calcium from the extracellular fluid. Those calcium channels concentrated in calveoli, or pockets on the sacrolemma. There are different channels gated by different stimuli that can respond to mechanical stretch, voltage, or chemical ligands. There's no troponin. Calcium instead binds to calmodulin protein, and then calmodulin activates myosin light chain kinase, which adds phosphate to the regulatory proteins on the myosin head. The thick filaments pull on the thin ones, and the thin ones pull on the dense bodies and the membranous plaques. Contraction and relaxation is very slow in comparison to skeletal muscle. The latent period in smooth muscle is 50 to 100 milliseconds. Tension peaks at about 500 milliseconds. declines over 1 to 2 seconds. That's due to slow myosin ATPase enzyme and slow calcium pumps. The latch bridge mechanism is a resistance to fatigue. The heads of the myosin molecule do not detach from the actin immediately, but they don't consume any more ATP. That can maintain a continual tonic contraction, which we refer to as smooth muscle tone. Stretch can open mechanically gated calcium channels in the sarcolemma, causing contractions. We see this in peristalsis. Stress relaxation response helps hollow organs gradually fill, like the urinary bladder. And unlike skeletal muscle, smooth muscle contracts forcefully even when greatly stretched. Smooth muscle has plasticity, the ability to adjust its tension to the degree of stretch. It's able to contract forcefully when stretched because there are no Z discs. The thick filaments will play a role, the thin filaments, in their arrangement versus the striations. Muscular dystrophy is a group of hereditary diseases in which the skeletal muscle degenerates and weakens and are replaced with fat and fibrous scar tissue. Duchenne muscular dystrophy is caused by a sex-linked recessive trait. Its prevalence is about 1 in 3,500 live-born males. Most common form, diagnosed generally around 5 years of age. The mutation in the gene for the muscular protein dystrophin and actin is not linked to the sarcolemma, so the cell membranes are damaged during contraction. We get necrosis and scar tissue as a result. Individuals with muscular dystrophy rarely live past 30 years of age due to effects on the respiratory and cardiac muscles. It is currently incurable. Myasthenia gravis is an autoimmune disease in which antibodies attack the neuromuscular junction and bind to acetylcholine receptors, putting them together in clusters. So the muscle fibers then remove those clusters of receptors from the sarcolemma by endocytosis, and the fiber becomes less and less sensitive to acetylcholine. The effects usually first appear in facial muscles. We get drooping eyelids and double vision due to strabismus, inability to fixate on the same point with both eyes. Can lead to difficulty swallowing and a weakness in the limbs. Typically occurs in women between the ages of 20 and 40. Can be treated with drugs to reduce acetylcholine breakdown and suppress the immune response. Can be tested by having a patient look upward for a given period of time, and you'll begin to see eyelid drooping complete by 90 seconds. All right, that wraps up